For this topic, we will be doing the same procedure as for the last two. We'll go over the process of electron transport chains, then I will be asking you questions that you should know for the exam. First, a bit about electron transport chains. When we combine hydrogen and oxygen gases with a little added energy to overcome the energy of activation, we get an explosion and water. As you can imagine, that would be bad for the cell. The cell, instead, releases energy slowly, generating a lot of ATP along the way. It's much more economical that way and less damaging. Here's another way to think of it. Which would you rather have next to your house? A forest fire or a lot of bonfires with firemen handy? So the cell slowly releases energy from the hydrogen through the process of passing its electron from carrier to carrier. Each carrier in the electron transport chain, as it is called, uses the energy of the electron to pump protons to the other side of a membrane. But we will discuss that in the next slide. Once the electron is all worn out, wrung of its last bit of usable energy, it is passed to oxygen. Oxygen, which now has a negative charge, picks up two hydrogen ions, or protons, to form water, and no boom. In eukaryotes, the ETC for aerobic respiration, or cellular respiration, is embedded in the cristae of the mitochondria. In prokaryotes, it is embedded in the cell membrane. Using the energy from the electron transport chain generates ATP. Inorganic phosphates are smushed directly onto ADP, so it is not substrate level phosphorylation. It is called oxidative phosphorylation because oxygen is involved. This is why you need to breathe in oxygen. And what do we exhale? That's right, CO2. So where does the CO2 come from in this process? That's right, from the citric acid cycle. So here is the electron transport chain in mitochondria. NADH brings a hydrogen to the first electron carrier in the chain. This electron carrier strips the electron from the poor little hydrogen and boots the poor electron-less proton into the space between the two membranes of the mitochondria. We call that the intermembrane space. Then the electron is passed to the next carrier. FADH2 comes along and gives two hydrogens to the next carrier. The electrons are passed from carrier to carrier until, all worn out and tired from its journey, the electrons are passed to oxygen. Oxygen is called the terminal or final electron acceptor. The oxygen accepts the worn out electron and picks up a few hydrogens that haven't been pumped out yet. Let's talk pH for a minute. Remember, the more hydrogen ions in the solution, the lower the pH, or more acidic the solution is. The fewer the hydrogen ions, the higher the pH, or the more basic. So which side of the membrane has the more acidic or lower pH solution in this picture? That's right, the dark orange side, which represents the inner membrane space of the mitochondria. So all these protons want to get to the other side of the membrane. It's crowded in the inner membrane space, but they can't get past the phospholipid bilayer of the membrane. Protons have a positive charge, and the phosphate heads have a negative charge. They kind of stick to each other whenever they get close. So, how do they get in where they want to go? They go through an enzyme called APT synthase. ATP synthase opens up a channel for the protons to go through. As they do so, the head of ATP synthase turns, smushing ADP and inorganic phosphate together. This makes a lot of ATP, between 32 and 34, depending upon if it's a eukaryotic or a prokaryotic cell. This is oxidative phosphorylation. This buildup of proton force on the other side of the membrane, which powers ATP synthase, is called chemiosmosis. Once again, I want you to know the why, the where, the starting substrates, the energy used, the energy produced, and the ending products. Come to lecture with lots of questions. I love answering questions.